Hello, am I audible? I have muted the microphones and cameras so that I can record the session without any problem. So, can you please let me know if I am audible? In the chat box, you can let me know if I am audible, and also let me know if you can see the video. Okay, thank you, Pratishtha. Let us just wait till it is 6 p.m. Then we will start today's session. Okay, do not leave the call. Please wait. We will start shortly once more students join. Can you let me know if you can see the presentation? Can you see the presentation which the PPT? Okay, thanks for confirming. We will start shortly once more students join. At 6 p.m. we will start. Okay. Till then, please wait. Very good evening to the students who have joined. Please wait. We will start at sharp at dot 6 p.m. Meanwhile, let me know if everything is all right with my voice, with the video, and also let me know if you can see the presentation correctly. You can use the chat box and you can then let me know if everything is all right with the audio, video, and also with the presentation. I hope I am audible. We will start at 6 p.m. Till then, please wait. Okay, thanks for confirming. Okay, we will start at 6 p.m. Meanwhile, you can just wait. I have muted your microphones and cameras so that I can record the session seamlessly. This recording will be shared with you after the class. So that is why. But the chat box is always open. So if you have anything you want to ask me, you can use the chat box. Very good afternoon, very good evening to the students who have joined. Please wait till 6 p.m. Then we will start today's session. Meanwhile, please let me know if you have any doubt or any query. I hope my voice is audible to you, and also I hope that you can see my video and the presentation. We will start once more students join. Okay, so till then, please wait. Since I'm recording the sessions and I will be sharing this recording with you after the class, that is why I have muted your microphones and the cameras so that I can record the session without any problem. But there is a chat box open here, and I am monitoring the chat box every time. So if you have any doubt, any query, so you can let me know in the chat box. We will start in 5 minutes, so please wait. I am waiting for more students to join, and then I will start. Meanwhile, let me know in the chat box if you have any issue with the audio or the video or the presentation. Okay, we will start in 5 minutes. Good evening, and again, I welcome you all. We will be starting our class shortly. Please wait. Okay, so I think we can start with the introductions. Meanwhile, I think most students will join, but with the students who have joined, I will start introducing myself and also I will start introducing the course in general. So, very good evening to all who have joined this class. I welcome you all in this NPTEL course of Organic Chemistry in Biology and Drug Development. This is a problem solving session. So, basically, and today it is a week one problem live session for this course. Okay. So, what is this problem solving session is all about? So, you know this, you have enrolled in this NPTEL course of Organic Chemistry in Biology and Drug Development. And this course, if you check in this the course catalog, you will find that this is a 12-week course, right? And each week, a certain portion of the course is released with the lecture and all the materials. And you have to attend all the lectures and which are readily available into the SWAM portal. And also, there will be quizzes every week, okay, and based on every week's course content. This class is basically to help you regarding the course material, the things, the topics which are taught in every week, and also this course, this live session or this problem solving session is intended to help you in your quiz solving the quizzes of the weekly quizzes, right? So one thing first going to our problem solving session, I would like to show you some few things. Okay, first I have already mentioned it earlier also, but since most students have joined later, I will tell you one more time that I am recording this session. Okay, because many students they cannot join the class on time due to some many problems. That is why I have to record the session for the students who cannot attend, and this recording will be shared to you after class in few coming few days. Okay, that is why I have muted your microphones and also the camera so that I can record the class without any problem. But as you can see, the chat box is always open here and I am monitoring the chat box every now and then. This chat box we will use to communicate during our classes. Okay. At the end of the session, at the end of the class, I will share my email ID with you. So if you have any doubt or any query regarding the class or in general regarding the course topics, you can let me know through the email also. Okay. I will share my email ID at the end of the session. And so if you go to the SWAM portal in the Organic Chemistry and Biology and Drug Development, you will find there is a left panel. From this panel, you can download the book from here. Okay. And also, if you click on a particular week's course content, you will find there is a quiz where you have to submit your assignment. All right. And in this left panel itself, there will be a new tab which will be updating in the coming days, which will say like problem solving session. From that portion, you can download the class, this recording, which I am recording the today's class, and also the upcoming classes. You can also download from that particular link, and also you can download the presentation which I will be sharing today. Okay, this presentation. So this thing will be updated in coming days, so you don't have to worry. As I have mentioned that today it is our week one live session. So before starting the session, I would like to clarify few things, and all these things are about what we'll be doing in our class on regular basis, and also there will be some general informations. Okay, so first let me start with this all the general discussions about the class. So I hope my voice is audible to you, and I also hope that the presentation is visible. If not, please let me know in the chat box. So there will be a problem solving session every week, and these sessions will be on every Friday from 6 to 8 p.m. So like today it is Friday, and we have starting our first lecture. The upcoming or more 12, 11 to 12 weeks, we will be having this problem solving session every Friday from 6 to 8 p.m. And these sessions are live on Google Meet platform. This is the link. You can write down the link anywhere you want, or also you will be reminded every before every class, one day before. Like, since our classes are on Friday, you will be reminded by the NPTEL site that there is a class on Friday on Thursday. Okay, the day before the class, you will be reminded. Or there also you will find this Google Meet link which you can use to join. Otherwise, you can write it anywhere you want. Okay. So one important thing is, like, since this is a live session on Google Meet platform, there can be some kind of network disruption. Okay, and if the network disruption happen, please do not leave the class. If somehow I got disconnected, I will rejoin as soon as possible. Okay, the students, you do not leave the class. And if there is any problem regarding your network, if you have to leave the session for some time, it is fine. The session is open. You can rejoin at any time. Okay, and since I will be sharing the recording with you, so you can check whatever you have missed during the poor network later also. During the session, you can post your doubts, your query, everything to the chat box. I have already mentioned it earlier also, that since I am recording the session, since I want to record it without any problem, that is why I have muted your microphones, but the chat box is always open, and you have any doubt, any query, you can let me know through the chat box. Okay. And still, if the doubt persists after class, you can drop an email to me. I will be sharing my email ID at the end of the session. You can use that email ID and you can drop your queries through the email also. And this presentation and recording will be shared to you. I have shown you where you will be finding it in coming days. Okay, just wait. After, by next week, it will be updated and you can record, you can download the presentation and you can see the class recording from that SWAYAM website itself. Okay. So I hope there is no problem till now. If you have any problem, you can let me know. Okay. So what? So one more thing, what we will be doing in our problem solving session. Okay. So this is the very important thing. So as I have already mentioned that will have to submit the weekly assignments. If you have checked already, there is a weekly assignment here. This assignments will be updated you with the email. This also you will get the email notification regarding also this. If you click on the assignment, you will find that it is a MCQ based assignment. So, okay. So you will have to answer this, and there will be a deadline which is mentioned here, and every assignment is marked. You have the points for answering in the assignments which you have to do. So what we will be doing in our problem solving session. So basically, in problem solving session, I will show you around 15 to 20 questions regarding each week's course content. So this is the week one live problem solving session, and today we will be discussing the week one course content, and also we will solve 15 to 20 MCQ questions. Okay, so what we will do, I will show you a particular question which will have 4 different options. Okay, you have to read the question carefully, and in the chat box you have to let me know A, B, C or D, which of the following will be the correct answer for that particular question. After showing you a question, I will wait for 5 to 6 minutes or 2 to 3 minutes. I will wait, and then I will check the chat box, and then I will show you the correct answer, and that topic I will discuss in very detail. That's how we will practice how to solve this kind of MCQ questions, which will give benefit you for solving the weekly assignments, and also it will help you to understand the course in general. Okay, and since it is a problem solving session and it is a practice session, there is no marks, okay, for answering during the questions. So you can answer with your free mind. There is no marks, so no negative marking is there and nothing. But I want all of you to participate. Okay, get involved in the class, participate and answer each question, even if your answer is wrong. It does not matter, since I am not going to mark you for that particular answering, but participation is very important. Otherwise, I won't understand that you are listening or not, or it will also help you to understand the course better. Okay. So now let us start with our first question of today's session. But before that, let us see what is in the week one course content. So This is the week 1 course content of our NPTEL course of Organic Chemistry in Biology and Drug Development. So the course contents are as follows: a brief introduction to molecules of life, structures of amino acids and their various charged forms, biological macromolecules and small molecules, importance and functions, amino acids, the building block of protein, amino acid separation and detection, electrophoresis and anhydride reactions. So these all are the content of your week 1 for this course of Organic Chemistry and Biology and Drug Development. Okay, so now let us start with our question answering session. As I have mentioned, I will show you the question there, which will have 4 different options. Read the question carefully and let me know in the chat box which of the following will be the correct answer for a particular question. After I check the chat box, after I will check your answers, then I will show you the answer and we will discuss that particular topic in detail. Okay. If you have any doubt, any query or any in general problem regarding audio, video or anything, please feel free to let me know in the chat box. So the first question of today's session, it says that match the amino acid with the side chain system present in it. Your options are: A is histidine, B is tryptophan, C is arginine. So these are the amino acids name, and the side chain system, number 1 is guanidine, number 2 is indole and number 3 is imidazole. Now you have to match the amino acid with the side chain system present in it. Your options are: number A has A with 2, B with 3, C with 1. Number B is A with 1, B with 2, C with 3. Number C is A with 3, B with 2 and C with 1. And number D is A with 2, B with 1 and C with 3. So this is a mix and match kind of question. There are 3 amino acids and 3 side chain systems are mentioned in the question. You have to identify which amino acid is going with which side chain system. There are 4 options: A, B, C and D. I will wait for 2 minutes. Then I will check the chat box and I want all of you to participate and answer. Even if your answer is wrong, it does not matter since I am not giving you any marks for it. But please try to answer the questions. Match the amino acids with the side chain system present in it. A is histidine, B is tryptophan, C is arginine. 1 is guanidine, 2 is indole and 3 is imidazole. Number A, option number A says A matches with 2, B matches with 3 and C matches with 1. Number B, A with 1, B with 2, C with 3. Number C, A with 3, B with 2 and C with 1. And number D, A with 2, B with 1 and C with 3. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 1. Let me check the chat box. Let me check if there is any answers. Okay, I think I have got many answers. Let me now check which of the following will be right. Okay, number C, number C, number C, number D, number D, number C, number A, number C. So I am seeing many number C, one number D and one number A. Okay, very good. All of you are answering, which is very good thing. So let me show you which of the following will be the correct answer and let us discuss about it. The correct answer will be, yes, it is number C. A matches with 3 means our histidine, it has imidazole ring. B matches with 2 means tryptophan has indole ring. C matches with 1 means arginine has guanidine ring. Now let us see what is the structure of the amino acids. This is the structure of histidine and this blue one is the imidazole ring system. This is the structure of tryptophan. This pink one or is the indole ring system. And this is the arginine and this red one is the guanidine ring system. So amino acid, the structures of amino acids are very important. and I mean, almost, if you cannot remember all of them, few of them you, I think you need to remember. Okay, the special amino acids which have this heterocyclic groups, the side chain, acidic, basic. So basically, amino acid, we classify the amino acid based on the side chain, based on the what type of side chain is present there. Okay, so sometimes the side chain can be non-polar and aliphatic. Okay, just like this one, these all, like glycine. The glycine has a side chain, only hydrogen is present there. There is no carbon containing side chain in glycine. So this is the glycine is the most simple amino acid, followed by alanine. Alanine has only a methyl. Then we have this valine group. Then we have leucine. Valine and leucine, you can see both are almost same. Only the problem, the change is that it has one carbon extra. Then we have methionine. Methionine is a special amino acid because it has a sulfur present here. And then we have this isoleucine. Isoleucine and leucine has a similar structure. Only the orientation is different. Okay, but the number of carbons are in the side chain are the same. Then we have the polar and polar side chain, but there is no charge present. Okay, so what are the examples? There is serine, there is threonine, which has this hydroxyl group, OH group. We have the cysteine, which has a thiol group, SH group. We have proline, which has a nitrogen containing ring. Okay, then we have asparagine, which has a CONH2 group, and we have a glutamine, which has similar structure with asparagine, only a carbon extra. Then we have the amino acids, we have this polar side chain, and the polar, it can be positively charged or negatively charged. The positively charged amino acids are the glycine, arginine and histidine. All these have the positively charged side chain which has a nitrogenous center. That is why these are the known as the basic amino acids. And for the negatively charged amino acids are the aspartate or glutamatine or aspartic acid glutamic acid, which has this acidic side chain also, which can stay as a carboxylate ion, which is negatively charged. And the amino acids which are have the aromatic side chain like phenylalanine. Then we have this tyrosine and tryptophan. So all these are the examples of amino acids, and these are the classification based on the side chain, whether the side chain is polar, non-polar, whether it has a positive charge or negative charge or aromatic side chain. Okay, so these things you have to remember. Okay, so any doubt till now? If there is any doubt, please let me know. Okay, I think the first question was all right, and most of you have given the correct answer. So very good that you are answering. Now we will move to the next question. Okay. Now problem number 2 says, which of the following statement is correct? Your options are: number A, alanine is not an essential amino acid but arginine is. Number B, glycine is an essential amino acid that has sulfur. Number C, valine is not an essential amino acid and it is acidic in nature. Number D, histidine is an acidic amino acid which is essential. So all the statements, there are four different statements, and you have to read the statements very carefully and let me know in the chat box A, B, C or D which of the following statement you think is correct one. Again, I will repeat. Number A, alanine is not an essential amino acid but arginine is. Number B, glycine is an essential amino acid that has sulfur. Number C, valine is not an essential amino acid and it is acidic in nature. Number D, histidine is an acidic amino acid which is essential. Read the options very carefully and let me know. Whenever I have seen most of the times that students make mistake in this kind of questions which are like correct, incorrect, true, false type of questions. That is why it is very necessary that you read the options very carefully. Again, I will wait for 2 minutes and again I want all of you to answer. Okay, in the chat box, let me know A, B, C or D which of the following is the correct answer. You don't have to worry because there is no negative marking. So answer whatever you think is the correct one. Which of the following statement is correct? Number A, alanine is not an essential amino acid, but arginine is. Number B, glycine is an essential amino acid that has sulfur. Number C, valine is not an essential amino acid and it is acidic in nature. Number D, histidine is an acidic amino acid which is essential. Okay, let me check the chat box now. Let me see how many of answers I have got. Okay, I got A is the correct one, and also I have got YA is the correct one. Very good. Then A, number D, number D, number D, number D. So most of the people think it is number D. Only I have got two number A. Okay, let me check which of the following is correct one. Let us find out. So there is a two options, number A and D. I have everyone has a doubt. Which of the following? So let us see. Number A is the correct answer. Okay. One student has written, and it is very correct, that alanine is not an essential amino acid, but arginine is. Number D is not correct because histidine is not an acetic amino acid. It is a basic amino acid. I have mentioned you earlier that histidine has this imidazole ring, right? And which has a positive charge, and this nitrogen is a this group ring is a basic in nature. So whatever. So these are the positively charged, like histidine, arginine, and lysine are the basic amino acid. Although histidine is essential, but it is not an acidic amino acid. Okay, so portion of the statement is not correct, so I cannot say that this statement is completely correct. Number A is completely correct, that alanine is not an essential amino acid, but arginine is an essential amino acid. Number B is not correct, it is incorrect. Lysine is neither an essential amino acid nor it has sulfur. And number C is also not correct. Okay, so first let us understand what are the essential amino acids. So basically an essential amino acid is an amino acid that cannot be synthesized itself by the organism, usually referring to humans, and therefore must be supplied in the diet. 10 amino acids are known as the essential amino acids. These are the arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Okay, these 10 amino acids, I hope you will memorize this because it is important. Okay, now remember the 10 amino acids which are essential. These amino acids must obtain from the diet. An adequate diet must contain these amino acids. Typically, they are supplied by meat or dairy products. Essential amino acids help the body function, regulate the neurotransmitters, chemicals in the brain that control the mood and behavior. A lack of essential amino acid can cause emotional and physical difficulties that lead to the health disorder. Okay. So this is a shortcut by which you can remember the name of this 10 amino acids, which says that any help in learning these little molecules proves truly valuable, where each and every letter is representing one amino acid. A represents arginine, H is histidine, I is isoleucine, L is leucine, T is threonine, L is lysine, M is methionine, P is phenylalanine, T is tryptophan, and V is valine. This is a shortcut that you can use to remember these things. Okay. So again, I am showing you the structures of the amino acids. Okay, amino acids with the electrically charged side chain, positive and negative. Positive ones are the basic amino acids, are the arginine, histidine, and lysine. Negative one are the acidic amino acids, which are the aspartic acid, glutamic acid. Amino acids with the polar uncharged side chain, serine, threonine, asparagine, glutamine. Special cases are the cysteine, glycine, and proline, because these are the special amino acids. And then amino acid with hydrophobic side chains are alanine, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, tryptophan. Whereas phenylalanine, tyrosine, tryptophan has this aromatic side chains. Okay. I hope all is clear till now. If there is any doubt, please let me know. Otherwise, I will proceed with the next question. Okay. Let me go to the next question. So problem number 3, it says that match the amino acids with the one letter codes. So every amino acid has a 3-letter code, also a 1-letter codes. Okay, 3-letter codes are relatively easier to remember because it is nothing the first 3 letters of the particular amino acid, like okay. But this 1-letter codes are sometimes a little difficult to remember, but it is very necessary. Okay, why it is necessary? I will coming in the upcoming slides. I will tell you why it is necessary. So first, match the amino acid with the 1-letter code. As the amino acids are, number A is asparagine, number B is tryptophan, number C is arginine, number D is lysine. And the 1-letter codes, number 1 is W, number 2 is R, number 3 is N, and number 4 is K. Okay, so match number A, option number A says A matches with 4, B matches with 3, C matches with 2, and D matches with 1. Number B, A with 1, B with 4, C with 3, D with 2. Number C, A with 3, B with 2, C with 4, D with 1. Number D, A with 3, B with 1, C with 2, and D with 4. I will wait for 2 minutes. Check and then let me know. Match the amino acid with the one letter code. A, B, C, and D are representing 4 amino acid. 1, 2, 3, and 4 are representing the one letter codes. There is any doubt? Okay, okay. So what is the doubt? There are 20 amino acid or say block building proteins. Why these are essential? You have mentioned why others are not. Okay. So what is okay, I will just I will answer your doubt first. Then others also please do the things. Okay. You can stop raising your hand. I have seen your doubt. So your doubt is there are 20 amino acids or say the block building proteins, and why only these are essential? You have mentioned why others are not. Okay. So when in the second question itself, I have mentioned you that essential amino acid are the amino acids that are not synthesized by our body. Okay. The other amino acids which I have not mentioned as essential, that amino acids are synthesized by our body itself. That need not to be taken by the diet. Okay. But the essential amino acids with the 10 amino acids which I have mentioned, these are essential amino acids because these amino acids you have to take through the diet. You will not find this amino acid synthesized in your body. Okay. That is why these are known as the essential amino acids. Okay, Asmin, I think. So, was your doubt got clarified? I cannot follow what is the clarification. I mean, which question? Number two, essential amino acid. Okay, I hope you have understood. Question number three. Yeah, question number three. I have not shown you the answer till now. So, okay, any answers? Let me first check if there is any answer for question number three, and then let me come to the question number three. I have not discussed it yet. Okay, so please wait. I will discuss it now. I have got one answer, number D. Number D also there is one another answer. Okay, I have got two answers which are saying it is number D. First, I will show you, then I will get I will give you the clarification regarding this problem number three. Okay. So, the correct answer for this problem number three is yes, number D. Okay. So, what now? Let me just discuss what is the one letter codes for the amino acids. Okay. So, amino acids. Again, here also it is mentioned what is the essential to diet and synthesis in human body, which is the essential amino acid and non-essential amino acid things are also mentioned in this table. Okay, so amino acids, to denote the amino acids, it is not possible for us to always write the complete name of amino acid. Okay, sometime especially when we are writing the chain of the amino acid, which we will discussing in our upcoming weeks. Okay, in protein or everything, it is not possible always to denote an amino acid by its complete name because sometimes it is not possible to write a complete name. That is why we use shorthand notations to denote a particular amino acid. Generally, these are the three letter codes and one letter code. Okay, one letter codes are more vastly used. Okay, especially in case whenever there is a topic matter of the this particular protein chain making or everything. Okay, we use this one letter code very often. We also use this three letter code. So the three letter codes are very easy actually. Okay, because it is the only first three letters of amino acids. For ALA for alanine, ARG for arginine. For asparagine it is ASN. For aspartic acid it is ASP, because both have ASP in the first. So for asparagine we have removed this ASP with ASN. For cysteine, it is CYS. For glutamic acid, it is GLU. And for glutamine, it is GLN. For glycine, it is GLY. Proline is PRO. For serine is SER. Tyrosine is TYR. For histidine, it is HIS. For isoleucine, it is ILE. For leucine, it is LEU. For lysine, it is LYS. Methionine is MET. For phenylalanine, it is PHE. For threonine, it is THR, tryptophan is TRP, and for valine, it is VAL. Okay, so these are the three letter codes of amino acids. And one letter codes, most of the cases, these are not only the first letter. Okay, sometimes we have to use the different letter to denote an amino acid, because sometimes it is the same letter is there, so we have to use. So why it is this particular letter, I don't know, because it is that is how it is denoted. It is not, there is any logic behind it. Okay. So alanine has denoted by A, arginine is denoted by R, asparagine is denoted by N, aspartic acid is denoted by D, cysteine is denoted by C, glutamic acid denoted by E, glutamine is denoted by Q, glycine is denoted by G, proline is by P, serine is by S, tyrosine is by Y, histidine is by H, isoleucine is by I, leucine is by L, lysine is by K, methionine is by M, phenylalanine is by F, threonine is by T, tryptophan is by W, and valine is by V. Okay, so these are the one letter codes that are for the particular amino acids. Okay. Okay, I hope you have understood the question number 3, right? So is there any doubt? Because somebody has doubt in the question number 3. If there is any doubt, please let me know. Okay, originally we can get the pure form of this amino acids in lab. It is you can buy it from the chemical. I mean the companies which sell the chemicals. So they also purify the amino acids. They synthesize, purify the amino acid, and they sell. Okay, so it is possible to get the pure form of the amino acids in lab. We can synthesize it. Okay, we can synthesize it. We can purify it, and also you can buy it from the vendors which sell the chemicals to the labs. Is there any doubt till now? Three questions we have discussed. Is there any doubt? Otherwise, I will move on to the fourth question. Okay, I hope you have understood the problem number 3 also. So basically, you have to remember these codes. Okay, most of the codes are easy to remember, because most of the codes are basically the first letter, but some codes you have to remember, like the arginine, it is different. For tyrosine, it is different one. So you have to just remember some things. Okay. Now I will go to the next slide. Okay. Problem number 4. Which of the following is the shorthand notation of metencaphaline? So it is the metencaphaline. This is the structure of metencaphaline. You have to identify what will be the shorthand notation of this pentapeptide. And the shorthand notation will be using your this one hand notation which I have mentioned in the last slide. Number A is YAAFM. Number B is YGGFM. Number C is FMMYG, and number D is FAAMG. Try to figure out what should be the first shorthand notation for it. Is there any doubt? I think somebody is raising hand or something. Okay, all this thing I will discuss in our upcoming session, so please don't worry about it. Okay, all these things I will discuss about amino acids. We have to lot of things we will discuss. Okay, this thing I will discuss at the end of the session. First, let me finish all the questions, then we will discuss. Okay, okay. Don't worry, we are only today, it is our first class. We will discuss a lot about the amino acids and proteins and so on. Let's focus on the problem number 4 and try to solve it first. What will be the shorthand notation for metenkephalline? What should be the shorthand notation for metenkephalline? I have mentioned earlier in early last slide what is the shorthand notation of which amino acids. If you have paid attention to the structures of amino acid, I think you can answer this question. Okay, let me check first if there are any answers for this question. Okay, I think for I got one answer, number B. Okay, let me show you what is the correct answer for this problem number 4. Very good. Whoever has answered it, I think I have received only one answer regarding this problem number 4, and I think that answer was number B. Yeah, very good. It is number B. So now let me show you why first hand amino acid. That is why the first hand notation or one letter codes of amino acids are very important, because it is very most of the time whenever we are discussing about a long chain of peptide or a long chain of amino acids, we use this one letter codes, okay, as a shorthand notation, because it is easy to write. So correct answer will be B. Now why? Well, let us understand first what is the met enkephalline. So this is the also known as the methionine enkephalline. It is a natural endogenous opioid peptide that acts as an agonist at opioid receptors. What is the agonist? What is the receptor? What is all these things we will be discussing in the upcoming classes. Okay, these are the vast topics which they will take time. So you don't have to worry about the terms which you will be seeing in today's class. You will be understanding the things after few classes. Okay, so it is very fine. It is one of the two forms of enkephalline. One is being the leuenkephalline. So now this is how you have to figure out what is the side chain. So this is a particular CONH. It is a peptide bond. Regarding peptide bond, what is the peptide bond and how it is formed, everything we will be discussing in our next class itself, I hope so. And then if you can see, this is the peptide bond. So this is the first amino acid chain. Okay, this is the first amino acid, and this is the tyrosine amino acid. If you remember, this is the tyrosine, the side chain, it itself it denotes the tyrosine, and this is the backbone. Okay, the CONH2, it is the backbone of the protein, and this is the side chain, which is the tyrosine. So tyrosine, the shorthand notation for tyrosine is Y. Okay, so Y is there. Then it is glycine. As you can see, there is only two hydrogen. There is nothing. No carbon is present. So this is glycine. So this will be G. This is also glycine. So G. This is the phenylalanine. As you can see, this is the side chain, the phenylalanine. So this is F for phenylalanine. And then we have SME, which is the methionine. So this is the complete 5 amino acids present in this pentapeptide: tyrosine, glycine, phenylalanine, methionine, and the shorthand notation will be YGGFM. So is this particular question number 4 is clear to you, or do I need to repeat it? So is the problem number 4 is clear? Did you understand the problem number 4, or do I need to repeat this thing? Let me know if it is not clear to you. I will repeat it one more time. You have understood how to identify what is the shorthand notation for a particular peptide. If the structure is given, how you will identify? If you have doubt, let me know. If not, we will move forward. I hope it is clear. Anyways, I will share this presentation with you after the class. Okay, yeah, thank you for letting me know. You don't have to worry. I will be sharing this presentation after the class. So if you have not understand this thing, you can practice it by yourself a few times after getting the PPT, and I think it will be okay. Otherwise, let me know. I will discuss it in the next class also. Okay. Now let me go to the next problem. Problem number 5 says, what are the 21st and 22nd amino acids that may be incorporated into the protein? Okay, so number A is the selenocysteine and pyrrolysine. Number B is selenocysteine and hydroxyproline. Number C is the hydroxyproline and pyrrolysine. Number D is none of the above. We know there are 20 amino acids, among which 10 amino acids are essential and other 10 are not, because the 10 we have to take by diet. We cannot synthesize it in our body. But there are two amino acids which are known as the 21st and 22nd amino acid, which may be incorporated in the proteins. You have to identify these 21st and 22nd amino acids. Your options are number A selenocysteine and pyrrolysine, number B selenocysteine and hydroxyproline, number C hydroxyproline and pyrrolysine, number D none of these are the correct answer. I will wait for 2 minutes. Let me know. A, B, C or D, which of the following you think is the correct answer? Let me know. Okay. So what are the 21st and 22nd amino acids that may be incorporated into the proteins? Let me check the chat box. Okay. We could have solved this question next week after we attended the classes for the same topics. So I think the course content is already given to you, right? The week 1 course content. See, this particular problem solving session, the thing is like the course content is released on every Friday. So as I hope you have got the email regarding the course content of week 2 is already live today, because I think I have got the email like this. Okay, like this. Okay. So I think course content is released on every Friday, right? And so you have a complete week, one Friday to Friday, to go through the course content, and after that we have this kind of problem solving session only to check if you have understood the course content correctly or not. But first you have to go through the course content, which is released on every Friday. And the week 1 course content, I think it is released okay, that is fine. You will go through it and it is also already for your benefit. Okay, I will share these things so you can go through the slides at any time. Okay, just attend the classes and I think it will be fine. Since you are enrolled today, you will find you will find all, yes, everything you will find in the SWAYAM portal. I have mentioned it earlier also. Everything is so today week 2 course content is released, and next Friday we will discuss the week 2 course content. That is why you have complete a week to go through the slides or go through the lectures. Okay. Okay, so for problem number 5, I have got three answers and two different options, B and A. Okay, let me check which of the following will be the correct answer. So the 21st and 22nd amino acids will be, number A is the correct answer, selenocysteine and pyrrolysine. Okay, these are the 21st and 22nd amino acid that may be incorporated into the proteins. So this is the structure of the selenocysteine, which is the just exactly same as cysteine, only in place of sulfur we has a selenium. Write this, okay? And this is the pyrrolysine. So pyrrolysine is that similar to lysine, but we have a pyrrole ring here, okay, present. So selenocysteine, or it is has a symbol SEC or U, is an okay. So it is the 21st proteogenic amino acid. Selenoproteins contain selenocysteine residues. Selenocysteine is an analog of more common cysteine with selenium in the place of sulfur. Selenocysteine is present in several enzymes, for example, glutathione peroxidases, tetraiodothyronine 5' deiodinases, and so on, formaldehydrogenases, glycine reductases, and so on. It occurs in all three domains of life, including important enzymes that are listed below present in the humans. Selenocysteine was discovered in 1974 by the biochemist Teresa Stedman at the National Institute of Health. Pyrrolysine, which has a symbol of PYL or O, can be encoded by the stop codon UAG, is a proteogenic amino acid that is used in some methionogenic archaea and in bacteria. It consists lysine with a 4-methylpyrrolidine 5-carboxylate amide linkage with the end of the lysine. It is a pyrrolidine side is similar to the lysine, is being basic and positively charged at the neutral hydrogen. Okay. So this is the structures of the selenocysteine and pyrrolysine, which are the 21st and 22nd amino acid, which may be incorporated into the proteins. Okay. So any doubt regarding the questions or till now? Okay, I hope it is clear. Then I will move to the next problem. So problem number 6, it says that which of the following will be the correct PI of glycine? PI means isoelectric point. The pK1 of glycine is 2.34, pK2 of glycine is 9.60. Your options are number A 7.26, number B 5.97, number C 3.63, and number D 4.11. Which of the following will be the correct PI of glycine? Let me know. A, B, C or D. After I check your answer, I will discuss what is isoelectric point and how to calculate the PI. That I will also discuss in detail. So whoever have not gone through the lecture slides till now, you don't have to worry. Okay, after attending the class, you can go through the lectures and you can correlate the things. It is very easy, so you don't have to worry about anything. I will give you 2 minutes, then I will check the chat box. Okay. Which of the following will be the correct isoelectric point of glycine? Given the pK1 of glycine is 2.34 and pK2 is uh, 9.60. Let me check the chat box. So let me check if there is any answer. Okay, I got many answers. Number B, number B, number B, number B. Okay, the formula is also given in the chat box. Very good. So you have gone through the lectures very well, which is very good thing. So let me discuss with the students regarding what is the isoelectric point. Let me discuss here. So the correct answer will be, yes, it is number B. So now let us first discuss what is the isoelectric point and how to calculate. So typically amino acids stays in the zwitterion, right? So amino acid which has a amine group and a carboxylic acid group. The amine group generally stays in the plus form and the carboxylate group in the negative form. Okay, so it generally stays as NH3 plus and COO minus. So which is known as a zwitterion. Zwitterion means we have the both positive and negative charges in a in one compound in a one ion, so that is known as a zwitterion. Okay, so typically amino acids are zwitterions at the neutral pH, and internal acid base reaction between the basic amino group and acidic carboxylic acid group results in a salt with 2 point of charges but a net charge of 0. Typical amino acids have two acidity constant Ka, one for carboxylate acid and one for the ammonium salt. Okay, so the carboxylic acid, the acidity constant is known as K1, and for the ammonium salt, the acidity constant is known as the Ka2. Okay, so in acidic condition, what happens? The both the COO group and the amine group both are the are which have the protons. Okay, so it is the overall charge is positive, which is the net charge is +1, which is in the acidic condition. Then first Ka1 is reached, which is the deprotonation of the carboxylate group, and we have this zwitterion, and the neutral condition, the net charge is 0, contains both positive and negative charges. Then in after reaching the acidity constant 2, which is for the deprotonation of the ammonium NH, we have this basic condition. This is the anion which has a net charge of -1. Okay, so isoelectric point of an amino acid at a pH which bears the net charge of 0, means it is the pH when the amino acid, the particular amino acid will stay in this zwitterionic form, which will have the net charge of 0. For amino acid with a non-acidic or non-basic side chain, the isoelectric point can be calculated by averaging the two pKa values. Okay, so Pi will be the average of this pK1 and pK2. Okay, and this thing is, this will be by 2. Okay, one thing, one second. This is a, so this is a pK1 plus pK2 divided by, so this pK will be average of these two, which is divided by 2. Okay, so for acidic, basic and basic amino acid we have 3 pKa values. For Pi of an acidic amino acid is obtained by averaging most acidic pKa values, and Pi of the basic amino acid is obtained by averaging two least acidic pKa values. These things I will discuss in upcoming slides. Okay, the how to calculate the Pi of the acidic and basic amino acids. So in general, for glycine and for this kind of amino acids, for the which for the neutral amino acids, we have only two pK values, pK1 and pK2, and the Pi will be the average of these two. Okay, so for glycine, the value will be 2.34 plus 9.60 divided by 2, which is 5.97. So this is the Pi of the glycine when the glycine stays as the neutral form. Okay, so the correct answer is number B. So I hope it is clear. Yeah, so number B was the correct answer. You have only for the normal amino acids, which are the neutral amino acid, you only have to average the amino acids, the two pK values are given. Okay, you have to only average these things. And how to calculate for the acidic and basic group amino acids, we will discuss in our upcoming slides. Okay, so number B was the correct answer for problem number 6. Then we will go to the next problem, problem number 7, which says for the peptide as shown as below, each pK value values are mentioned. Okay, the isoelectric point will be number A 6.30, number B 2.52, number C 7.09, and number D 9.74. So this is the peptide. This carboxylate group has 1.88 pK value. This has 3.65. This have 8.95, and this have the 10.53. So now you have to find out what will be the isoelectric point of this particular peptide. Just think what is the isoelectric point. The isoelectric point will be the value when the particular peptide will be in neutral form. Try to calculate the isoelectric point of this particular peptide. I will give you 2 minutes and then I will check the chat box and I will show you how to do or how to solve this kind of questions. For the peptide as shown below, each pK values are mentioned. Isoelectric point will be, let me check the chat box. Let me check if there is any answer given for the question number 7. Okay, I have got a few answers. Number A, number A. So most of the answers I have got, which is saying it is number A. Let me check if number A is the correct answer for problem number 7. So the correct answer will be, very good, it is number A. So isoelectric point is the pH at which the peptide will have the neutral form. So as you can see, this particular peptide has plus this for this carboxylate acid which has the pK value of 1.88. Okay, so after 1.88 we will have this as a negative, right? And the overall charge will be plus 1 and minus 1, which is plus 1. At this pK value, when the pK value is 3.65, this will get negative value, and we will have this neutral form. And this neutral form, which started from the 3.65, it will continue up to 8.95. When this ammonium ion, it will lose a proton, and overall charge will get minus 1. Okay, so this, from this pK to this pK, the amino acid, this particular peptide stays in a neutral form. Okay, and after that it will get another minus 2 form. So that is why we have to average these two pK values between which this this particular peptide is staying as a neutral, and it will give you the pK value, which is 6.30. So yeah, number A is the correct answer, and very good. I have got almost many answers which are saying this is the correct answer. Yeah, very good. First, we have to check the zwitterionic form, which will be having the carboxylate group and amino acid. You have to find out what are the pK values to have this zwitterionic form, and you have to average it out. Okay. Anybody has doubt in this particular question? If you have any doubt for the particular question, please let me know. I will discuss it one more time. Otherwise, let me just move forward with the question number 8. Okay, if you have doubt, let me know. I will discuss it at the end of the session also. Okay, so problem number 8, calculate the PI value of glutamic acid provided pK1 is 2.19, pK2 is 9.67, pK3 is 4.525, where pK1 for the pK of the alpha carboxylic acid, pK2 is the pK for the alpha ammonium ion, and pK3 is the pK value of the side chain. So glutamic acid is the acidic amino acid. You have to now find out what is the PI value. I have mentioned when the amino acid is not acidic or not basic, which is normal, so there will be two pK values. You have to average the pK values, which will make this, which will give you the isoelectric point. But for the acidic and basic amino acid, we will having three pK values. Now you have to solve this thing. This is the similar what we have done in our last question. So I think now all of you can answer this question. I will show you in detail how to solve this question. First let me wait for 2 minutes. Write in the chat box after calculating what is the PI of glutamic acid. What will be the PI value of the glutamic acid provided pK1 is 2.19, pK2 is 9.67, pK3 is 4.25, where pK1 is the pK value of alpha carboxylic acid, pK2 is the for alpha ammonium, and pK3 for the side chain residue. Let me check the chat box. I think I will have more answers. Yeah, very good. You have gone through the slides very well. Very good. Okay, let me check. Number B. I have got two number B and one number D. I am not getting many answers, and I want all of you to participate even if your answer is wrong. You have to answer, okay? Otherwise, I won't understand that you are following the class or not. Okay, let me check which of the following will be the correct answer. So the correct answer will be number D, 3.22. Now, how to solve this kind of question? So this is the structure of the glutamic acid, right? So this is the structure when all the at the in acidic pH, when all the groups are the protonated. As you can see, when all the groups are protonated, we have the net charge of +1. Since it has an extra carboxylate acid group, okay, in the neutral, in the acidic condition, we will have a net charge of +1 at the pK 2.19. Now, when the at the pK 2.19, what is the pK for? It is the pK for the alpha carboxylate ions. So we will get this form which has the net charge of what? Net charge of 0. This is the deuterionic form. Then after in the pK 4.25, we will have this is the pK for the side chain. So we will have this form which has a net charge of -1, and in the pK 9.67, which is the pK of the alpha ammonium ion, we have the net charge of -2. So deuterion, as we know, is the ion which is the is neutral, and the PI value is the uh, average of the two pK values in between the deuterion is present. That is why we have to average these two values, 2.19 and 4.25, which is in the and the deuterion is between these two. So the deuterion is 2.19 plus 4.25 divided by 2, which is 3.22. So that is how you have to solve the PI questions of the particular acidic amino acid. So just remember, whenever there is an acidic amino acid, you have to average the lowest pK values. Okay, the lower two pK values you have to average for the acidic amino acids. Okay, so this is the correct answer for problem number 8. Now I will go to the problem number 9. Problem number 9 says calculate the PI value for the lysine provided pK1 is 2.18, pK2 is 8.95, pK3 is 10.53, where pK1 is the for alpha carboxylic acid, pK2 is for the alpha ammonium ion, and pK3 is the for the side chain residue. Your options are number A 2.18, number B 5.56, number C 9.74, and number D 6.35. So what will be the PI value for the lysine? The last example we have done for the PI calculation for the acidic amino acid. Now it is the time for the calculating the PI for the basic amino acid lysine. So that's how we have completed the PI calculation of the all three types of amino acid, the neutral one, the acidic one. Now we are going to calculate for the basic one. So this summarizes your PI topics very well. Okay. And I know now if you are given any question, you can solve it very easily. So what will be the value of PI for lysine? Provided the pK1 is 2.18, pK2 is 8.95, and pK3 is 10.53, where pK1 is for alpha carboxylic acid, pK2 is for alpha ammonium ion, and pK3 is the side chain residues. So just you have to do it just like last time. You have to draw the. I think this is very easy to if you draw the structure, and I think you will can do it very easily. I feel that. You can remember the formulas as well, which is very fine, but understanding the concept behind the formula is also very important. So let me first check the chat box. I think, or I will wait for 1 more minute, then I will check the chat box. Okay. So for this carboxylate ion, carboxylic acid group, the pK value is 2.18. For this one, this ammonium ion, the pK value is 8.95, and for this one, the pK value is 10.53. Let me check the chat box. If you have any doubt regarding this pK calculation or PI calculations, please let me know. Okay. Number B, number C, and it is number B. So I have got number D. Okay, I have got number B, C, and D, three different options as answer for this question. That is very fine. You, at least you are trying, which is good. And so let me show you. So for in case of basic amino acid, you have to average out the correct answer is number C. Okay. So why it is number C? Now let me show you. So this is the structure of this lysine at the neutral pH. Now you have one more ammonium group. In the last case, we had one more carboxylic acid group. So the net charge was only +1, but here we have one more ammonium group. So the now net charge is +1, +1, +2. At the pK of 2.18, we will get this structure when this carboxylic acid group, it will lose a proton and we get this carboxylate ion. So now the net charge is +1, +1, -1, which is +1. Then at the pK 8.95, which is the pK value for this ammonium ion, we will now this ammonium ion will get deprotonated and we will get the net charge of 0, only -1, and here we have a +1. Now the pK value at 10.953, what will happen? This will get deprotonated and we will have a net charge of -1 only for this carboxylate group. Okay, so what will be the isoelectric point? It will be average of these two pK values. In between the amino acid is getting the zwitterionic form. So we have to average this pK 8.95 and 10.53. So we get the value of 9.74, which is the option number C. So just remember, in case of acidic amino acid, we have to average this lowest to lower two values, and in case of basic amino acid, you have to average the higher two values. Is it clear, or I need to repeat it? Yeah, this PPTs you will get. I have already shown you at the in the beginning of the class. I think that you will find there is a your SWAM portal where there is a left panel, right, where the weekly courses are updated. Here will be another tab which will be updated soon in upcoming 2 to 3 days, which will show you the problem solving sessions. If you go to that particular tab, you will get this PPT and also you will get the recording of today's class in case you have missed anything due to the poor network connection. Okay, so you don't have to worry. You will get the class PPT and this. Okay, regarding the exam and all these things, I will discussing in our upcoming classes. Okay, just let us finish about 2 to 3 classes, then we will go into the get into the examination and all the stuff. Okay, you don't have to worry about it. We will discuss it in due time. So, any doubt in question number? I think 9. Otherwise, I will move to the next problem. Okay, I hope there is no problem. Then we will discuss we will go to the next problem. So problem number 10, it says that identify the incorrect statement. Your options are number A, in the interior of the globular protein, hydrophobic amino acids are present. Number B, hydrophilic amino acids stay in the interior of the globular protein. Number C, valine will reside on the interior surface. And number D, globular proteins are water soluble. Identify the statement which is incorrect. The problem number 10, number option, your statement number A, it says that in the interior of the globular proteins, hydrophobic amino acids are present. Number B, hydrophilic amino acids stay in the interior of the globular protein. Number C, valine will reside on the interior surface. And number D, globular proteins are water soluble. Which of the following statement is incorrect? Read the options very carefully and then let me know in the chat box A, B, C or D which of the following statement is the incorrect regarding the globular proteins. We will discuss regarding the globular proteins and all these things in our upcoming class itself. I think in the week 2 class also we will discuss about it. The what is the globular protein and all those things. But this question is related to the amino acids itself. So that is why I have given it today. Just read the options and let me know whatever you think is the correct answer. Your answer, if your answer is wrong, it does not matter. I have already said you multiple times. So please feel free and answer. Which of the following will be the incorrect statement? Number A, in the interior of globular proteins, hydrophobic amino acids are present. Number B, hydrophilic amino acids stay in the interior of the globular protein. Number C, valine will reside on the interior surface. Number D, globular proteins are water soluble. So, which of the following is the incorrect statement? Let me check the chat box. Okay, so I think I have got. Okay, for the neutral amino acid, it is very easy because the neutral amino acid you will have only two basically pK values, right? I have mentioned it for the glycine itself. As you can see for glycine or for the neutral amino acid, you will have only two pK values and you have to make the average of it. pK1 plus pK2 divided by 2 will give you the isoelectric point. Okay, it is very easy for the neutral amino acid. Only two pK values, you have to make the average of it. Okay, for I think for the question number 10, I have got few answers and all are saying it is number B except one I have got now as a number A. Okay, let me check. Okay, where was I? Okay, so the statement which is incorrect will be number B, hydrophilic amino acids stay in the interior of the globular protein. It is not correct. Okay, so first let us understand. So I have discussed it in a couple of times, so I will skip this thing. So basically, the amino acid, one thing I will just sorry. So the amino acids which have the neutral side chain or which has the non-polar side chains, these are considered as the hydrophobic one, and the amino acid which has the polar side chain or charged side chains, these are considered as the hydrophilic one. So what are the globular proteins? So globular proteins are named for their approximately spherical shapes and are most abundant protein in the nature. The globular proteins exist in enormous variety of three-dimensional structures. Nearly all the globular proteins contain substantial number of alpha helices and beta sheets into the compact structure that is stabilized by polar and non-polar interactions. Globular proteins are three-dimensional structure formed spontaneously and is maintained as a result of the interaction among the side chains of the amino acid. Most often, the hydrophobic amino acids are buried closely packed in the interior of the globular acid out of the contact of water, and hydrophilic amino acids lies on the surface of the globular protein exposed to the water. Consequently, globular proteins are usually very soluble in the aqueous solutions, and diversity of the protein structures reflect in the remarkable variety of functions performed by the globular proteins like binding, catalysis, regulation, transport, immunity, and cellular signaling and more. Okay. So basically, globular proteins have this particular hydrophilic amino acids in the interior of exterior of the say in the surface. Okay, but that is why these globular proteins are water soluble, and in the interior it has this hydrophobic amino acids. So now let's go through the options. So number A, it says the interior hydrophobic amino acids are present, which is correct statement. Number B, hydrophilic amino acids are the interior of globular protein. This is not correct. Hydrophilic amino acids will be staying on the exterior of the globular protein, making these proteins water soluble. Number C, valine will reside on the interior surface, which is also correct because valine is a non-polar or hydrophobic one, and globular proteins are water soluble. This is also correct. So the incorrect statement is number B. Okay. So now I will go to the next problem. Problem number 11 says, which of the following statements is true? Your options are: number A, all amino acids have S configuration. Number B, cysteine is the only amino acid that has the R configuration. Number C, methionine is the only amino acids having the R configuration. And number D, both cysteine and methionine have the R configuration. Which of the following statements is true? Your options are: number A, all amino acids have the S configuration. Number B, cysteine is the only amino acid that has the R configuration. Number C, methionine is the only amino acids having R configuration. And number D, both cysteine and methionine have the R configuration. Which of the following statements is true? Let me know in the chat box A, B, C or D, which of the following you think is the true statement. Which of the following statements is true? Your option: number A, all amino acids have the S configuration. Number B, cysteine is the only amino acid that has the R configuration. Number C, methionine is the only amino acids having R configuration. Number D, both cysteine and methionine have the R configuration. Let me check the chat box. Let me check if I got any answer for the question. Okay. I think this is the answer. Number B, number B, number B. Okay, so all of you think that number B is the correct answer. Now let me check if number B is the correct answer or not, and let us discuss about it. So the correct answer will be yes, number B, cysteine is the only amino acid that has a R configuration. So basically, the RS notifications are like this. This is the structure of cysteine, and this is, as you can see, this is the above the plane bond, and this is the below the plane bond. Okay. So basically, we tried to. So these are the numbering based on the priority of the group. So generally, in general, so whenever there is a normal, in case of normal amino acid, when there is no sulfur present, the priority goes like 1, 2 and 3. Okay, and which goes to the anti-clockwise direction, and we have the S configuration. But only thing is that for sulfur, we have for cysteine, we have this SH. The bond is directly connected to the S, which has the second priority, and it has this. If you go to 1, 2, and 3, we will give the clockwise rotation. We will give us the R configuration. So this is the exam. This is the table to find out which will be the when it is R, when it is the S. So is the lowest priority group number 4 is going away from you? Yes. Here hydrogen is going away for us because it is the below the plane. So it is going away from us. If it is yes, then I side moves in the clockwise direction 1, 2, 3. It will have the R configuration. And if the I side moves from the 1, 2, 3 anti-clockwise, then it is the S. And if this hydrogen or the priority group number 4, it is above the plane. Okay, if it is above the plane, then what will be happen? The I side moves in the clockwise direction will give us the S configuration, and in the anti-clockwise will give us the R configuration. Here, priority group number 4, it is in the going away from us. So 1, 2, 3 clockwise direction will give us the R configuration, and cysteine is the only amino acid that has this R configuration. Okay. Now I will go to the next problem. Problem number 12 says, which statement regarding the drug thalidomide is true? Your options are: number A, R isomer is a sedative. Number B, S isomer is a sedative. Number C, both R and S isomers can be used as drug. And number D, R isomer is a teratogenic. Which of the following statements regarding the drug thalidomide is true? Number A, R isomer is sedative. Number B, S isomer is sedative. Number C, both R and S isomers can be used as drug. And number D, R isomer is teratogenic. Which of the following statements regarding the drug thalidomide is true? Option number A, R isomer is sedative. Option number B, S isomer is sedative. Number C, both R and S isomers can be used as drug. And number D, R isomer is teratogenic. Let me know A, B, C or D, which of the following is the correct answer regarding thalidomide. I will wait for 1 minute, then I will check the chat box and we will discuss it in detail. Okay, let me check the chat box. Let me check if I got any answer. Okay, Asmin, regarding the examinations, I will discuss it in upcoming weeks. It is only the first week, so you don't have to worry. We have till 12 more weeks to go, then we will have the examination, right? I will discuss in due time regarding the questions regarding asked in the examination in the final examination, okay? So you don't have to worry about the final examination right now, okay? Okay, so for the next questions, I have got the answers like number A, number D, number A, number C, number A, okay, number A, C, and D. These are the different options which I have got for the problem number 12. So regarding the final examination and everything, I will discuss in the due course of time. It's only the first class, so you don't have to worry about it. We will discuss it in after some weeks, okay? Because it takes some time to change and the question pattern and everything. Sometimes it gets modified. So in due time, we will discuss about it. So you don't have to worry about it right now. Okay, so I think I have got few answers, number A, number C, number D. I have got, I think, three different options as answers. So let me check which of the following is the correct answer for problem number 12. The correct answer will be number A, R isomer is a sedative, okay? And S isomer is a teratogenic, and both cannot be used as a drug because S isomer is not safe to use as a drug. Okay, so these are the structure of the thalidomide. So this is the R thalidomide and this is the S thalidomide, okay? Thalidomide was originally marketed as a safe, non-toxic sedative and antiemetic in the 1950s and rapidly became the popular to counter the effects of the morning sickness during pregnancy. Unfortunately, it was instrumental in one of the major medical disasters of the modern times when it is produced teratogenic effects in developing fetuses and lead to the babies being born with the stunted limbs and other development deformities and so called the thalidomide babies. As thalidomide was considered as such a safe drug at the time, few suspected that it was the cause of the problem, and by the time it was linked to the deformities, it was withdrawn in 1967. 8,000 to 12,000 babies were affected. Consequently, thalidomide gained a lasting notoriety when it comes to instrumental in the significant tightening of the regulations surrounding the testing of the drugs. Despite its notorious past, there has been the continuing interest in thalidomide and it has some remarkable properties which indicate a wide variety of clinical uses. Early on, it was recognized that thalidomide has an anti-inflammatory property which was useful in treating the leprosy. This activity was eventually linked to the thalidomide's ability to inhibit synthesis the pro-inflammatory endogenous cytokine TNF alpha, which is produced by the monocyst. In 1998, the drug was approved for the treatment of leprosy. However, thalidomide has a raft of other properties. For example, it is an immunosuppressant and could possibly be used for the treatment of autoimmune diseases or in countering the immune response to allow the organ transplants to be accepted by the host. Interest in thalidomide as an anti-cancer agent started when it was found that it inhibited the angiogenesis by unknown mechanism. Tests showed that thalidomide did indeed have anti-cancer activity, and it entered the phase 3 clinical trials for the treatment of renal cancer and multiple myeloma on that basis. Since then, it has been discovered that anti-cancer properties of thalidomide are more complex than its effects on the angiogenesis alone. In some patients, thalidomide can boost the immune system by variety of mechanisms rather than suppress it, and it is too many account to anti-cancer activity. As thalidomide can suppress and boost the immune system depending on the individual circumstances, it is known to as the immunomodulator. It appears to arrest the growth of the cell and promote the apoptosis directly. Okay, so the analogs. So basically, this case, what happened during the this thalidomide. So first, the thalidomide, which was sold as a drug, was the racemic mixture of this plus and minus this R and S thalidomide, the racemic mixture. Okay, so what happens in the R form is a sedative, which is good for the I mean, which was the intended use of the thalidomide. This S form was the teratogenic effect. So that is the actually a very good example how the stereochemistry can affect the effect of a particular drug and why pure compound or pure drugs, pure enantiomers are necessary. Okay, so these are the analogs of thalidomide that has been synthesized with the aim of removing the teratogenic properties. Lenalidomide, this is the structure. Okay, which is known as the Revlimid and Actimid. That are two such examples. Both contain an amino acid substituent on the aromatic ring, which was found to be crucial for the producing safer drug. Revlimid entered the clinical trials in the year of 2000, was given orphan drug status in 2001 for the treatment and then incurable disease of multiple myeloma. In 2003, it entered the phase 3 clinical trials and was given fast track status, and now it appeared for the treatment of multiple myeloma. Okay, so okay, immunosuppressant mean it actually lowers the immune response. Okay, we have all immunity and everything. So some actually components they actually suppress this particular immunity, and it is a one mechanism to have some kind of drug and some kind of diseases. These things I will also discuss in our upcoming weeks. Okay, in detail. Today, let's not discuss about it because we don't have much time left, and I have to complete more questions. So we will discuss these things more in detail in the next classes. Okay. If the time permits, at the end, I will discuss what is the immunosuppressant mean. Okay. So I think it is the question is clear. I will discuss your doubt at the end. Okay. So first, let me finish the more questions. Then we will discuss it. Okay. So problem number 13, it says that which of the following amino acid fails to produce the blue color in the ninhydrin test? Your options are number A arginine, number B tyrosine, number C proline, and number D asparagine. Which of the following amino acid fails to produce the blue color in the ninhydrin test? Arginine, tyrosine, proline, or asparagine? Let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 13? Which of the following amino acids fail to produce the blue color in the ninhydrin test? Arginine, tyrosine, proline, or asparagine? Let me know A, B, C or D. Which of the following amino acid fails to produce the blue color in the anhydrine test? Arginine, tyrosine, proline or asparagine? Okay, let me check the chat box. I think I will get some answers. Number C, number C, number C, number A, number C. Okay, so most of the answers I have got the number C, I think, and one answer I have got as the number A. Let me check which of the following will be the correct answer. And then let us discuss about the what is an anhydrine test. So the correct answer is yes, it is number C, proline. Proline is the only amino acid that fails to give the blue color in the anhydrine test. So basically, anhydrine test is a very good test. I mean, good indication or indicator test for the amino acids if it is present. It will if you test the amino acids with the anhydrine, it will produce a bluish purple color, which is known as the Rueman's purple. Okay, so what is the mechanism? So this is the structure of the anhydrine, as you can see, and this is your amino acid, right? So first, now what will happen? This intermediate amine will form, and we will have this intermediate amine, and will have this aldehyde and carboxylic acid. Okay, and then this intermediate amine will react with another molecule of anhydrine to produce this bluish purple color. Okay, but in case of proline, what happens? Proline is not like our simple amino acid. So what happens in case of other amino acids? This NH2 group is free. This alpha NH2 group is free, and this free alpha NH2 group reacts with this particular groups and gives us this aldehyde and this intermediate amine. But in case of proline, this NH2 group is not free. Rather, it is a part of a pyrrole ring, and this is the only proline is the only amino acid which has this cyclic structure, and where this is where the amine group is not free. What is the mechanism with the reaction of anhydrine with the proline? So anhydrine can also stay in this form. Okay, so this lone pair of nitrogen attacks on this place, and we will get this, and then after getting protonated we will get this type of structure, which will get an intermediate transfer of this proton, and release of carboxylic acid will get this type of zwitterionic structure, this positive negative conjugation, and this structure is responsible for the brownish yellow color produced by the proline in presence of anhydrine. Okay, so anhydrine not only it fails to produce the bluish purple color in presence of proline, it produces a brownish yellow color, and this is the structure because of which this particular anhydrine test of proline gives us the brownish yellow color. Okay, for this structure. So is it understandable, or is there any problem? I have got the correct answer in the chat box. Most of you have said that it is number C. Okay, if it is all right, then I will move towards the next problem. Okay, now problem number 14 says, which of the following amino acid will produce the yellowish orange color in the xanthoprotic test? Your options are number A arginine, number B proline, number C aspartic acid, and number D tryptophan. Which of the following acids will produce an yellowish orange color in xanthoprotic test? Arginine, proline, aspartic acid, or tryptophan? So we have several tests, color tests for the detection of the amino acids. Anhydrine test is one. We have a xanthoprotic test and so on. We have a few more amino acid detection tests present. So now you have to identify which of the following amino acid will produce yellowish orange color in the xanthoprotic test. Arginine, proline, aspartic acid, or tryptophan. Which of the following amino acid will produce yellowish orange color in xanthoprotic test? Which of the following amino acid will produce yellowish orange color in xanthoprotic test? Arginine, proline, aspartic acid, tryptophan. Let me check the chat box. Okay, number D, number D. I have got most as a number D. One number B. Okay. Also, I have got nice explanation for option number D. Okay, let me check and let me show you the correct answer. So the correct answer will be yes, it is number D, tryptophan, and yes, it is correct that xanthoprotic test, the aromatic amino acids basically give this yellowish orange color in the xanthoprotic test. So what is the principle? The test is used to identify the presence of the aromatic amino acids like tryptophan and tyrosine in the protein sample. The sample is treated with the nitric acid. The formation of the yellow color indicates the presence of the aromatic amino acid. The test is based on the fact that tyrosine is heated with nitric acid. It undergoes the reaction to produce a yellowish pigment called xanthoprotein. Not only tyrosine, tryptophan and phenylalanine also, in presence of this nitric acid and 40% NaOH, it will produce this nitrated amino acids which will give this yellowish orange color. So xanthoprotic test, the definition, principle, and result. So it is if test is negative means the absence of the aromatic amino acid tyrosine and tryptophan, and absence will of the dark and yellow orange color. And the xanthoprotic test is positive, the presence of the aromatic amino acid tyrosine and tryptophan, the presence of the dark and yellow and orange color. Okay. So yeah, number D was the correct answer. Now problem number 15, it says which of the following statements is true regarding the movement of amino acids in the gel electrophoresis. Your options are number A, if pH is less than Pi, then it will move toward the negative electrode. Number B, if pH is less than Pi, then it will move toward the positive electrode. Number C, if the pH is greater than Pi, then it will move toward the negative electrode. And number D, if the pH equal to Pi, then it will move toward the negative electrode. Which of the following statements is true regarding the movement of amino acids in the gel electrophoresis? Read the options carefully and then let me know A, B, C or D, which of the following will be the correct answer. Number A, if the pH less than Pi, then it will move toward the negative electrode. Number B, if pH is less than Pi, then it will move toward the positive electrode. Number C, if pH greater than Pi, it will move toward the negative electrode. And number D, if pH equal to Pi, then it will move toward the negative electrode. Which of the following statements is true regarding the movement of amino acids in the gel electrophoresis? I will wait for a minute, then I will show you the correct answer and we will discuss it after checking the chat box, obviously. So please try to answer the question. Which of the following is true regarding the movement of amino acids in the gel electrophoresis? We already have discussed how the amino acids will behave during the in course of the pH and PIs. We have already discussed. Let me check the chat box, okay? So for problem number 15, do I have any answer? Number A, number A, number B. So I have number A, number D, number B. So I have three different options. I have number A, number B, and also number D as the answers. So let me check which of the following will be the correct answer. So number A, number B, and number D, three different options I have got. So let me check which is the correct answer. The correct answer will be number A. Okay, if pH is less than Pi, then it will move toward the negative electrode. Now, why? So gel electrophoresis separates amino acids based on their net charge and size, more accurately charge to mass ratio. Okay, now when an electric field is applied across the gel, charged amino acid migrate toward the oppositely charged electrode, and the rate of the migration is influenced by the amino acid's charge and size, with smaller and more highly charged amino acid moving faster. Amino acids have ionizable groups, amino and carboxylate group, plus any ionizable group on their side chains that can gain or lose proton depending on the pH of the solution. This determines the net charge of the amino acids. At the pH below the isoelectric point or pi, the amino acids will have a net positive charge and migrate towards the cathode, the negative electrode. At a pH above their isoelectric point, amino acids will have a net negative charge and migrate toward the anode or the positive electrode, and at the isoelectric point amino acids will not have any net charge and it will not migrate. Okay, so remember, if pH is below the isoelectric point means pH is less than pi, the amino acid will have a net positive charge and it will migrate to the negative electrode, and the pH is greater than pi means pH is greater than pi, then it will make the amino acid will have a net negative charge and it will migrate to the positive electrode. Okay, so number A is the correct answer. Now let me go to the next problem. Problem number 16, it is based on the last question itself. Which amino acid will move toward the negative electrode at pH 6 during the paper electrophoresis? Your options are number A, glutamic acid, number C, B, cysteine, number C, lysine, and number D, methionine. Which amino acid will move toward the negative electrode at pH of 6 during the paper electrophoresis? Your options are number A, glutamic acid, number B, cysteine, number C, lysine, and number D, methionine. Is there any doubt? But number B is answer, because higher pH, the molecule to gain negative charge as a hydroxide ion. Number B option is lower pH than isoelectric point. Okay, let me just check once. So in case of the lower pH, the particular amino acid, it will have a positive charge. If you remember the last questions which I have discussed earlier, is you can see in the lower pH, when the pH is less than pi, say in case of, let's say in case of this lysine, the pi value is 9.74. So if the pH is, let's say the pH is less than this 9.74 value, this particular amino acid, it has a positive charge, and if it has a positive charge, then it will move to the negative electrode, and after the pi, if the pH is greater than pi, the particular amino acid will have a negative charge and it will go to the positive electrode, right? I do not understand what is the query is about. At the isoelectric point, the particular amino acid stays as a zwitterionic form, right? So if the pH is less than pi, what was the particular answer? I think if the pH is less than pi, then the particular amino acid will stay as a positively charged ion and it will migrate to the negative electrode. That is the problem number A, right? The option number A. Because if it is the positive charge, then why it will go to the positive electrode? Is it clear or not? Just remember the example of the any amino acid, I will say, at the pi, like let's say for the pi, for the glutamic acid, the pi is 3.22. At this pH is basically, pi is basically the pH at which the amino acid stays in the zwitterionic form when the net charge is 0. So it will not migrate at any electrode. But if the pH is less than, let's say the pH is 1 or pH is 2, then what will happen? If the pH is less, there is more H plus protons, means the amino acid is completely protonated and has a positive charge, then it will go to the negative electrode. Is the doubt clear, or there is still the doubt? Anyone else who have doubt in this question? If the pH is greater than pi, the particular amino acid, when the pH is greater than pi, the amino acid is staying in the negative charge and it will migrate to the positive electrode. That is written here also, right? I think you should go through the slide when I will share it, okay? And if the doubt persists, I will discuss it in the next class. Let me go to the next question. And let me show you the what is the answer. So which amino acid will migrate to the negative electrode at pH of 6? That was the question number 16. And the correct answer will be number C, lysine. So what is the paper electrophoresis? So paper electrophoresis is a separation technique mainly used to separate the amino acids and protein and peptides. Separation of mixture based on the charge and size of the molecule. Migration of the charged particles takes place during the through the paper under the influence of electric field. The paper strip is used must contain at least alpha cellulose or a percent of 95% or should have a slight absorption capacity. Paper does not contain any charge that interfere with the separation of the compounds. So basically, I have already mentioned if pH is less than isoelectric point, that will be present in the positively charged species and will migrate to the negative electrode. That is, I have mentioned in the last slide also, right? That if pH is less than pi, the amino acid will have the net positive charge and it will net positive charge and it will migrate to the negative electrode. And if pH is greater than pi, the amino acid will present in the negatively charged species and it will migrate to the positive electrode. Now the question says which of the following amino acid will move toward the negative electrode at pH of 6, means at the pH of 6 it will be positively charged, means its pi must be greater than 6. So the PIs of lysine, cysteine, glutamic acid and methionines are respectively 9.74, 5.07, 3.22 and 5.74. Only for lysine the PI value is greater than the pH 7. Okay, that is why means the pH is less than the PI. That means lysine will present as the positively charged species and will move toward the negative electrode. Is there any doubt? Yeah, number C was the correct answer. See, you go through the slides, you go through these things. These things might sometime you may feel that this is a little bit problematic or little bit confusing. But once you go through it, I think it will be I mean, you will understand these things. Okay. If not, please let me know through the email. I will share my email ID. I will discuss it in the next class itself. If you have any doubt regarding this isoelectric point and this migration of the amino acid to the electrode at a particular pH, this topic, if you have doubt after today's class and after going through the slides, I will discuss it in the next class also. Okay, just let me know through the email that I have to discuss it in the next class. Now let us go to the next slide. So problem number 70 says in the primary metabolism of our body, proteins are controlled by what? Your options are number A carbohydrates, number B nucleic acids, number C lipids and number D enzymes. So in the primary metabolism of our body, proteins are controlled by which of the following? Number A carbohydrates, number B nucleic acid, number C lipids and number D enzymes. In the primary metabolism of our body, proteins are controlled by which of the following? Carbohydrates, nucleic acids, lipids or enzymes? So what will be the correct answer for problem number 17? Let me check the chat box because we don't have much time and I have more 2 to 3 questions left. Okay. number D, number B, number B. So I have got mostly number B and one number D. I have got the answer. Okay, let me check which of the following is the correct answer for this problem number 17. So, in the primary metabolism of our body, proteins are controlled by, very good, number B, nucleic acids. So, this is the primary metabolism of our body. Okay, we have this in plants, we have this photosynthesis which uses this and this photosynthesis will give us the glucose, and we have this pyruvic acid acetyl CoA citric acid system. So this is a little complicated, just only for your understanding, you don't have to memorize it, just I have given it for your understanding. Okay, then for glucose we have these sugars, we give this polysaccharides and also the riboses. This ribose sugar and all these are connected with the phosphates with ATP also. This ATP also is connected with the amino acids, and also we have this nucleotides. Okay, and this nucleotides gives us the nucleic acids, which stores the genetic information. Okay, and this controls the proteins. Okay, now this nucleic acid controls the protein, and proteins control as enzymes the all chemical reaction in our body. Okay, so nucleic acids control the protein, proteins control all the chemical reactions as a form of the enzyme. Okay, so single-headed arrow this represents basically the chemical reaction in the usual sense, starting material incorporated into the product, like nucleotides are incorporated into the ATP, and there is this dashed arrow, it's the compound needed for the reaction but not always incorporated in the product, like these things. Okay, so these are all the notations, we can go through it. So what are the differences between the nucleic acid and amino acid? So basically nucleic acid is a complex organic molecule such as DNA and RNA made up by many nucleotides linked with the long chain. Amino acid is a simple organic molecule which contains both carboxylic and amino groups. Nucleic acids are basically polymer, amino acids are monomer. Monomer is the particularly nucleic acids consists the monomer is the nucleic acid and polymer is the protein. Okay, protein this for amino acid is the monomer of the protein, which is the polymer, made up of carbon, hydrogen, oxygen, nitrogen and phosphorus. This nucleic acid, amino acids are basically made up of carbon, hydrogen, oxygen, nitrogen and sulfur. Nucleic acid is made up of pentose sugar, nitrogenous bases and phosphate group, and this amino acids contain carboxylic group and amino groups. Nucleic acid phosphodiester bonds occur, peptide bonds occur in amino acids. DNA and RNA are two types of nucleic acid. Proteins are made up of 20 amino acids synthesized inside the cell by DNA replication and transcription, either synthesized or obtained from diet. The nucleic acids store the genetic information of the cell and are involved in the synthesis of functional proteins, and amino acids used in the translation of the mRNA, the building blocks of the protein. Okay, so these are the differences or comparisons between the nucleic acids and amino acids. Now, problem number 18. Which of the following statements is true regarding the peptide bond formation? Number A, peptide bond is not planar in nature. Number B, the carbon-nitrogen bond is purely single bond in nature. Number C, the cis form is more sterically favored than the trans form. Number D, cis conformation is favorable only when the proline is present. So which of the following statements is true regarding the peptide bond formation? Number A, the peptide bond is not planar in nature. Number B, the carbon-nitrogen bond is purely single bond in nature. Number C, the cis form is more sterically favored than trans form. Number D, cis conformation is favorable only when the proline is present. Peptide bond formation and these things we will discuss in more detail in the next class, but I thought to give a one question to see if you have any idea about the peptide bond formation about these things. Try to answer this question. Which of the following statements is true regarding peptide bond formation? Let me check the chat box because we don't have much time and we still have two more questions left. Okay, number B, number D. I have two different answers. Let me check which of the following is the correct answer regarding the peptide bond formation. So the correct answer will be number D. Cis conformation is favorable only when the proline is present. This statement is true regarding the peptide bond formation. So peptide bond is basically the bond between two different amino acid. One carboxylate group of carboxylic acid group of one amino acid reacting with the amine group of another amino acid. So it's basically the you can say this is the amide bond formation. Since it is happens in protein, it is known as the peptide bond formation. So peptide bond formation is basically, so the first statement which says that it is the peptide bonds are not planar in nature, which is false, because this peptide bond, it is planar in nature. It is planar. It is this NH and this CO, this bond, it stays in the same plane. Okay, this is not a pure single bond in nature, because there is a, let me just show you. So as you can see, there is a lone pair of nitrogen participates in the resonance, and we have this double bond structures is sometime in between the carbon nitrogen bond. So it is not a pure single bond character. Also, this transform, this carboxylic and NH bond, they prefer to stay trans to each other, not to cis to each other, because if it stays cis to each other, there will be a steric hindrance. But only the cis formation, bond formation is favorable if the proline is present, because proline is a cyclic amino acid, right? So the peptide bonds in protein is planar in nature as the result of the resonance structure shown in here. This gives the peptide bond a significant double bond character, which prevents the rotation. As a result, bond rotation in protein backbone is only possible for the bonds on either side of the each peptide bond. This has an important consequences for protein tertiary structure. There are two possible conformation for the protein bond. The trans conformation is the one that normally present in the protein, and the cis conformation leads to the steric clash of the residues. However, cis conformation is possible when the peptide bonds is next to the proline residue. Okay, so number D, which says the cis conformation is favorable only when the proline is present, is the correct statement. Now, problem number 19. It says which of the following statements regarding lipid is not true. Your options: number A, it is the main component of the cell membrane. Number B, it includes fats, oils, steroids and waxes. Number C, most of the lipids are water soluble. Number D, lipids also have an important role in cellular communications. Which of the following statements regarding lipid is not true? Number A, it is the main component of cell membrane. Number B, it includes the fats, oils, steroids and waxes. Number C, most of the lipids are water soluble. Number D, lipids also have an important role in cellular communication. Which of the following statement is not true? I think it is very easy, and I know all of you can answer this question. I will wait for a minute and then I will check the chat box and show you the correct answer. Which of the following statements regarding the lipid is not true? I think it is very easy question and all of you can answer this. Let me so check the chat box. Lipids are insoluble in water. Very good. Yeah, number C. All of you have answered very well. Okay, so now let me show you and let us discuss a little bit. Then so yes, the statement which is not true is number C. Lipids are water soluble. No, lipids are not water soluble. So what are lipids? Lipids are the macromolecules which are made of the fatty acid monomers. Function of lipids include the structural support for the cell, energy storage and cell signaling. Lipids usually are non-polar in nature and do not interact with water, though some exceptions exist. Okay, so the lipid types are the triglyceride, phospholipids, steroids and waxes. So lipids are the principal component of the cell membrane along with the cholesterol, also the component of the cell membrane. They have acquired a bad name, but they are nevertheless essential for the function of the membranes as a selective barrier of the movement of molecules. The most common types of lipids are ester and glycerol. Glycerol is just propane 1,2,3 triol, but interestingly stereochemistry, it is not a chiral, it has the plane of symmetry, but the two primary OH groups are enantiotropic. It's one of the modified esterification. For example, the molecule becomes chiral. So, okay, and natural glycerol 3-phosphate is such an ester and that is optically active. This one. A typical lipid is in foodstuffs is the triester formed by the glycerol and oleic acid. So this is the structure of the oleic acid, and this is which is the most abundant lipid in the olive oil. Oleic acid is a mono unsaturated fatty acid that has a Z double bond in the middle of the C18 chain. This bond gives the molecule a marked kink that in the middle, and the compound is actually present in olive oil as a triester, which is also kinked. So this is the glyceryl trialloid, which is main lipid component of the olive oil, which has a this Z double bond in between the 9th and 10th carbon. Okay. So this is all about the lipids. And the last question of today's session, which of the following statements regarding the prostaglandins is false? Your options are number A, they are a class of steroid molecules. Number B, they are a group C15 carboxylic acids derived from the arachidonic acid. Number C, prostaglandins varies depending on the geometries and position of the double bonds. And number D, they regulate the kidney function and also control the inflammation. Which of the statements regarding prostaglandins is false? Number A, they are a class of the steroid molecules. Number B, they are a group 15 C15 carboxylic acids derived from the arachidonic acid. Number C, prostaglandin varies depending on the geometries and position of the double bonds. Number D, they regulate the kidney function and also control the inflammation. Okay, let me check the chat box and discuss because we are almost at the end of the session. Is there any answer? Number B, number A, number A. So I have got two different options as answer. Okay, number A and number B. Let me check. So the statement which is false is very good. It is number B, which says it is a group C15 carboxylic acids derived from the arachidonic acid. So basically, prostaglandins are the group of naturally occurring unsaturated hydroxyl or unsaturated or hydroxy keto fatty acids. Okay, so these are C20 carboxylic acids, not C15. That is why the statement number B is false. Which is with the cyclopentane ring. The cyclopentane ring with two side of chain contains usually a beta and a keto and enone hydroxyl functionalities. Okay, so prostaglandins are a group of lipid compounds known as the eicosanoids and have a diverse hormone-like effects in animals, including humans. They are produced in almost every tissue, play a wide range of roles in body, including regulating inflammation, cell growth, smooth muscle contraction. Prostaglandins are involved in processes like blood clot formation, blood flow, healing, and menstruation. And these are the C20 carboxylic acids, not the C15. Okay. So this was the last question of today's session. So today we discussed about the basic building blocks of the amino acid and all the things regarding amino acid like isoelectric points and the structures of amino acid. These things we have discussed today. Now I will share my email ID. If you have any doubt or any query regarding today's class, please feel free to reach me through the email. This presentation and class video will be uploaded to the SWAM portal. You have to just check it regularly. Within a week, within I think 2 to 3 days, it will be updated in the SWAM portal. Okay, the today's class and this lecture PPT. So if you have any doubt, any query, please let me know through the email. I will discuss that particular topic if you have any doubt in the next class also. Okay. And somebody has asked what is the immunosuppressant. I don't know the student is present here or not. So basically immunosuppressants are the compounds that suppress our immunity system. And now you may think why it is necessary, because sometimes the suppressing our immunity is necessary in certain cases. Like for example, I will give you one example. Like in case of the organ transplant. So if the foreign organ is transplanted in our body, our body will not take up that organ, okay, because it is the foreign particle. That time we have to give the immunosuppressant drugs so that the immune response are lower, so that our body can take up that particular organ. So this is the one example. There are several examples of the immunosuppressant drug. You can read it from the Google also. It is very easy thing, okay. And I will discuss in I think in our one of any classes. I think we will discuss it. So I think the doubt is clear. Somebody has asked regarding this immunosuppressant and I have told that I will say it last. Okay, I hope the student is present. So thank you all for joining. Today was our first class for this organic chemistry and biology and drug development, and we will have this kind of lectures every Friday from 6 to 8 p.m. I will close the session for today. If you have any doubt or any query, please feel free to reach me through this email ID. Okay. I will close the session for today. Thank you all. Thank you all. I am closing the session. Let's meet on the next Friday from this time. Okay.